Day 9 today is going to be about the cell cycle and mainly focusing on interphase and mitosis. So cells will grow and divide through this cell cycle, and we're going to get into the details of what each of the parts are and what the important things that happen in each of the parts are. So if you are on the Google Classroom post, make sure that you open up the cheat sheet or the one page of notes to help you work through the questions in the slide set and through the exit ticket. So the first part that we're going to talk about is interphase. That's where cell growth and DNA replication happens. The second part is going to be mitosis, where the nucleus of the cell is going to split and the DNA is going to be separated into two separate nuclei. And then the third part is going to be cytokinesis, where the actual cell and cytoplasm split up into two. So physically, there are going to be two cells at the end, each with their own nucleus. So inside of interphase, what are the three phases? Can you write them out for me? Just looking at this diagram, you can use it to help you. What are the three parts of interphase? Well, if you're looking at the diagram, interphase is actually the majority of the cell cycle. So over here, this little pie piece right here is mitosis, and everything else is interphase, as you can see from this arrow. And interphase has three main parts: G1, where we have normal growth and function; S phase, where you have DNA synthesis, right, where DNA is replicating or duplicating. S stands for synthesis. And then the third one is the G2 phase, where the cell is going to prepare to do mitosis right here. And it's double checking that during G1, S, and G2, everything has been duplicated, including the DNA and organelles. So during S phase, again, what is occurring? Is it A, DNA synthesis, B, DNA transcription, or C, DNA deletion? Pick your answer. The answer is that it is A, DNA synthesis. Remember again that the S for S phase stands also for synthesis of DNA, which is when the DNA is being replicated, and now there's going to be two sets or double the amount after you finish S phase. So cells will spend most of their time in interphase, like we represent in the cell cycle, right? Mitosis is just this tiny little chunk right here, and it happens really, really quickly compared to the rest of interphase or the rest of the whole cell cycle, because 90% of the time, the cell is going to be in interphase, doing growth, doing its normal functions, duplicating its DNA, doing its normal functions again, and growing a little bit more before it double checks everything to get ready for mitosis. And mitosis is just a really quick process of split, split, done. So why must DNA replicate during the S phase? Why is this so important? Well, DNA has to replicate, because if it didn't, then the cell would be dividing anyways in mitosis. And when it divides in mitosis, then the daughter cells that are supposed to be identical would actually only have half the number of DNA, or sorry, half the amount of DNA, or half the number of chromosomes that it would need, and it wouldn't be able to function, and the cell would immediately fail or die. So this is an issue that actually is possible to happen, but there's also checkpoints inside of the cell cycle to make sure that this doesn't happen, because that's a huge mistake and a waste of time to even try to duplicate the DNA or duplicate the cell if the DNA inside isn't even ready to prepare and be in two separate nuclei and be a full set of DNA. In mitosis, remember, the daughter cells are identical to the parent cell. So the DNA has to replicate, because each daughter cell must get a full set of DNA. We're not making sperm and egg here, we're making body cells. Your body cells need all 46 chromosomes. So if you start with 46, you got to duplicate them so that when they split, you can get 46 and 46 for each daughter cell too. So when it does replicate, or when DNA is synthesized, DNA replication is semi-conservative. Now, what does this mean? Well, let's look at the steps here. In step 1, the DNA double helix will unwind into ladder form through the use of the enzyme helicase. And the hydrogen bonds in step 2 between each base pair, ATGC, will break. Each strand of DNA will serve as a template for the new DNA to be created. And in step 3, the enzyme DNA polymerase will add complementary nucleotides to the original DNA strand or DNA template to form new DNA strands, right? They're matching now. Step 4, at the end, 2 DNA double helixes that are identical to each other will be created. And each is going to be made up of half old and half new. Okay, so semi-conservative is talking about how each strand is made up half old and half new of the DNA. That's what it means. Conservative would mean that it's completely old and completely brand new. And that's not exactly what happens, right? We don't have a strand that is completely light blue and completely dark blue, right? We also don't have any other version of this. So it's half old, half new DNA. That's why it's semi-conservative, because it's half and half. So after interphase, the DNA is replicated and the cell is ready to begin dividing. So it's going to enter mitosis if nothing is going wrong. Mitosis is where we experience the cell division, mainly of the nucleus, and then at the end there's cytokinesis that'll physically separate the cell. So if you do remember, let's just see if you do remember. I want you to drag the red dot to the image that represents prophase, the green dot to the image that represents telophase, the blue dot to the image that represents anaphase, and then the orange dot to the diagram that represents metaphase. So here are the actual 4 phases. Hopefully you labeled them right on the last slide. So the order that they happen in is prophase, metaphase, anaphase, telophase, so PMAT or PMAT for short. Prophase is the first one, and that's where the spindle fibers are starting to develop and the nuclear membrane is slowly dissolving, and you're preparing to do the rest of the steps that you're going to split the nucleus. Metaphase is where the spindle fibers have connected to all the chromosomes at the centromere or the centers, and they are lining them up along the middle or metaphase plate. Anaphase is when those spindle fibers are now going to be pulling the chromosomes apart by their sister chromatids, right? The A in anaphase stands for apart or away. So they're being pulled apart. And then telophase is when the 2 separate nuclei are going to reform. And so you have 2 nuclei. And then after telophase, you'll actually separate physically into 2 separate cells. So again, mitosis is PMAT, prophase, metaphase, anaphase, and telophase. Mitosis is only the nucleus division though. So prophase again is getting ready to divide. Metaphase, the chromosomes are in the middle. It's a very obvious thing to see if you're looking at pictures. Anaphase, the chromosomes are being pulled apart. So they used to be in the middle and now they're being pulled apart half and half to each side. And telophase is where you see 2 nuclei reforming. The membranes are reforming for the nuclear membranes. And then afterwards, you're going to have them actually split. So remember, like I keep saying, mitosis splits the nucleus. So that every cell, when it's dividing into 2, will have a whole nucleus with the correct amount of chromosomes. So here I want you to order and write out the 4 phases of mitosis using their full name. Don't just use their letters. Remember the acronym that we use to help? Remember the orders of the phases. Make sure you write out the full words. So the correct answer is PMAT, which represents prophase, metaphase, anaphase, telophase. If you just wrote PMAT, I can't give you points. If you also just wrote any other shorthand version, I can't give you points. You got to make sure you write out the full words so that you can help remember. But writing out the acronym PMAT here can help you then write out the full words of the 4 phases. So cytokinesis is the actual end of cell division. Mitosis divides the nucleus, but physically the cell is still one whole cell. It just has 2 nuclei inside of it. So when the 2 nuclei are actually fully formed, we have to divide the cell physically. And the cell membrane will pinch in and in and in, where there's myosin and actin fibers that are going to pull the middle part in and then in until it completely meets and then pinches apart into 2 separate cells. So it's kind of like if you pinch a piece of dough or pinch a piece of clay, the cell is also pinching in at the middle until it moves, it separates completely. So cytokinesis is the actual physical cell separation. It's called cytokinesis because the cytoplasm is moving, right? The division of the cytoplasm at the cleavage furrow or the middle where it's going to pinch in is what we call cytokinesis. So here's an image showing where it's pinching in, and then you're going to get the squished in very, very close until it actually breaks off and physically separates into 2 separate cells. Notice that the nuclear membranes are reforming as cytoplasm is starting because that's the end of telophase. In these pictures right here, we have a cell A and we have a cell B. Which 2 phases of mitosis are being represented by A and B in this microscopic image? And how do you know? Tell me, what is the identification that you can use to tell me what phase, prophase, metaphase, anaphase, or telophase that A is in? And B, is it in prophase, metaphase, anaphase, or telophase? How did you know? So A is anaphase. And the reason why I know that is because you can see the chromosomes being pulled to the opposite ends of the cell. Anaphase stands for apart, remember? So the sister chromatids are being pulled apart. On the other hand, B is metaphase, since all the chromosomes are lined up in the middle. Yeah, they're kind of messy, but they're pretty much all aligned along one single file line in the middle of the cell. So that would be the metaphase plate, or the middle. Between mitosis and cytokinesis, which one of these is in charge of splitting the nucleus? Drag the dot to the correct answer. Between mitosis and cytokinesis, which one of these is in charge of splitting the cytoplasm? This one should be pretty obvious, it's in the name. The nucleus is split during mitosis, and the cytoplasm is split during cytokinesis. Right? It helps that the name begins with cyto. So mitosis splits the nucleus during mitosis, obviously PMAT, and then at the end of PMAT, cytokinesis will happen so that the physical separation of the cell will occur, and you'll end up with two separate cells with their two full sets of DNA from mitosis. So what happens if the cell cycle actually messes up? Well, there are checkpoints that happen during the cell cycle to prevent damaged cells from duplicating. It's between G1 and S right here. Inside of S to make sure the DNA has replicated properly. And then there's one between S and G2. And then there's one between at the beginning of mitosis, right after G2. These are all important because if the cell is not properly done with the phase it was supposed to just have finished, then either it's going to stop and retry to do the phase, which was growth, duplicating DNA, duplicating organelles, splitting properly, or if it's already messed up so badly, the cell will just commit apoptosis, which is cell death, and then it's going to just restart with another cell to do cell cycle. Right? If the checkpoints don't catch a mistake, or they just aren't stopping the cell for some reason, then you can get uncontrolled replication of cells. And when cells keep replicating over and over and over and splitting and splitting and splitting and splitting, even when they're not supposed to, then you can get things like tumors and cancers, and they can be pretty serious diseases. Sometimes they're caused by mutations, sometimes they're just caused by your genetics that you inherited. So cancer cells, like I said, will ignore signals that should cause them to stop dividing. So they're going to divide over and over and over and over again, even when they're not supposed to. So when normal cells are grown in a dish, they're crowded by neighbors on all sides, so they will no longer divide. But cancer cells will keep dividing and pile on top of each other in lumpy layers, even when they're completely overcrowded, right? They don't recognize the idea that they're not supposed to keep growing, and they don't recognize any signal that also tells them to stop growing. So cancer cells have unregulated cell division. They will continuously go through the cell cycle, even if the cell has mistakes or mutations in it, and then they will cause issues to an organism's health because of this uncontrolled growth. So let's try a practice problem together as an example before you do some independent work on your own. We are going to use the RAKE method where we read and highlight, ask the question again, show our knowledge, and eliminate the answer choices that don't work and pick our best answer. So the question that we're going to read is, the diagram shows cells in different stages of mitosis. A student is trying to find a cell in a particular phase of mitosis. The student is looking for evidence that spindle fibers are separating the chromosomes to ensure that each new nucleus has one copy of each chromosome. Here are the diagrams. We have 1, 2, 3, 4. Notice that in 1, there are spindle fibers. They're lying along the middle. There are spindle fibers in 2, but they're being pulled apart. In 3, a cell plate is forming to, conjoin the cell wall and separate the two cells. And in cell 4, the chromosomes are formed, but the nucleus still hasn't broken down yet. So in this question, they're asking which cell is a phase of mitosis that the student is looking for. Is it 1, 2, 3, or 4? Let's highlight the important parts. We're looking for a particular phase of mitosis. So these are all in mitosis right now. And we are looking that spindle fibers are separating the chromosome. That's a huge key hint, right? We're looking for one of these where the spindle fibers are separating the chromosomes. Now, what is the question asking? Well, to combine the info from here and here into one good question instead of just this vague question that's not really good down here, we're going to say which picture shows spindle fibers that are in the process of separating chromosomes. So what knowledge do we have on this topic? Well, we know that we don't even need to know the name of this phase. We just need to know that spindle fibers are separating sister chromatids, and that's what we're looking for. So let's eliminate some answers, right? In answers cell 3 and cell 4, we don't even have spindle fibers, so we can automatically eliminate them, right? So pick what you think is the correct answer out of the left two, cell 1 or cell 2, and we'll see on the next slide if you get it right. So because we are looking for spindle fibers separating chromosomes, cell 1 is going to be incorrect because the fibers haven't separated them yet. They've touched and connected and moved them to the middle, but they haven't separated them yet. So cell 2 is going to be the correct answer. And if you were wondering, this is an example of anaphase because they are being pulled apart. This is metaphase, this is telophase or telophase, and this is prophase. So all the phases are shown here, but only 2 of them have spindle fibers and only 1 of them is pulling the chromosomes apart. So now you're going to work through some practice problems with RAKE. Here you have 1, 2, 3 problems to work through, and you're going to go ahead and finish those. Make sure you're doing all your steps before you go on to do your exit ticket.